Welcome to this video on investigating out-of-specification, or OOS, test results. This course is based on FDA guidance and outlines a structured approach to Phase 1 and Phase 2 OOS investigations. Our objective is to ensure data integrity, product quality, and ultimately, patient safety. Quality control is the pursuit of truth, not just compliance. An OOS result is test data outside established acceptance criteria. Importantly, rejecting a batch does not negate the need for an investigation. Root cause determination is mandatory under FDA regulations. The OOS investigation follows a defined life cycle. It begins with OOS detection, moves to a Phase 1 lab investigation, and then hits a decision gate. If no clear error is found, it escalates to a full Phase 2 investigation. Each gate ensures the process is controlled and scientifically justified. Phase 1 begins with the analyst's responsibility at the bench. Analysts must verify system suitability and immediately stop testing if an obvious error occurs. The golden rule is, do not continue testing to see what happens. All observations and errors must be documented promptly. The supervisor performs an objective assessment using a 7-point checklist. This includes examining raw data, verifying calculations, and confirming instrument performance. Hypotheses of lab error must be scientifically tested using retained samples whenever possible. Decision Gate 1 determines whether a clear laboratory error or assignable cause exists. If confirmed, the OOS result is invalidated. If no clear error is found, the default path is Phase 2. OOS results must never be attributed to analytical error without conclusive evidence. Phase 2 moves the investigation to the manufacturing floor, led by the quality unit. A cross-functional team reviews production records and process history. If a manufacturing root cause is confirmed, the investigation concludes. If not, the team proceeds to additional testing. Retesting analyzes additional portions of the same original sample. It must follow a predefined SOP for the maximum number of tests. Do not repeat testing indefinitely until a passing result is obtained, which is known as testing into compliance. Unsatisfactory retest results indicate a suspect batch. Resampling uses new material collected from the batch. This is only appropriate when the investigation proves the original sample was not representative or improperly prepared. Resampling is never justified, simply because the original result was OOS. Mathematics should be a tool, not a mask. Averaging results to hide variability or combine OOS with passing values is misleading and inappropriate. Individual test results must be reported and evaluated on their own merit. Outlier tests are informational, not exculpatory. They cannot be used to invalidate chemical assay results. Statistical exclusion does not prove laboratory error. Apparent outliers are often the result of real sample variability and should not be used to justify passing an OOS result. If the investigation confirms an OOS affecting batch quality, the batch must be rejected. If results remain inconclusive, the quality unit reviews all data. Release is only acceptable when evidence demonstrates the OOS does not reflect true batch quality. When in doubt, prioritize patient safety. For distributed product, field alert reports, or FARs, must be submitted within 3 working days of OOS discovery. This is required even if the investigation is incomplete. A follow-up FAR must be submitted when the investigation concludes. To summarize, stop analysis immediately if error is noted, retain all preparations, and use a second analyst for retesting. Do not test into compliance, discard samples prematurely, or use outlier tests to invalidate results. OOS investigations must be objective and scientifically justified. The goal of an OOS investigation is to determine the true quality of the product, not simply to pass the batch. Laboratory error should be rare. When uncertainty exists, the quality unit must prioritize patient safety and scientific integrity over batch release. Thank you for visiting at cGMP Training on YouTube. If your company needs customized training for your operation, please contact me at spandit@gmpscientific.com.